Hello everyone and welcome to today's webinar, Automation-Aided Targeted Genotyping with Allegro for Maize Breeding, a case study of QTL analysis with magic mapping populations. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by TCAN. To learn more, visit them at tcan.com. Now, we encourage you to participate today by submitting any questions you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speakers, Dr. Davide Scaglione, the Chief Technology Innovation Officer at IGATech Technology Services, and joining him will be Dr. Leonardo Caproni, a postdoctoral fellow at the Institute of Life Sciences at the Santa Ana School of Advanced Studies in Pisa, Italy. Welcome, gentlemen. You may now begin your presentations.
Hello, everyone. Thanks for joining this webinar. My name is Davide Scaglione. I'm the CTIO of IGATech, and today I'm going to present on automation-aided Allegro targeted genotyping for maize breeding, a case study for QTL analysis on magic mapping population. And today the presentation is going to be hosted by two. The first part is going to be on me. I'm going to present you more on the technology and the implementation of the automated platform. While on the second part, Leonardo Caproni from Scuola Superiore Sant'Anna will give better insights into the analysis of the data. So a very quick introduction to IGATech, the company I'm working for. We have as a main mission to provide technologies and methods based on sequencing to improve the health of human, animals, and our planet in general. And among those, of course, we foster the preservation of biodiversity and also the improvement of agriculturally relevant crops. Of course, to do that, not only in the agribiotech field, we need to maintain a quite wide portfolio of technologies and methods, and not only also analysis. And so we range from the novo assembly of plant genomes down to genotyping exercises, but also we are serving for epigenetic studies, metagenomic studies, and also some single-cell analysis of transcriptome and epigenome. After having said that, genotyping is one of the applications we are running the most because now with sequencing-based genotyping, the demand has increased quite a lot during the recent years. As you know, genotyping is the art of sampling the diversity in large cohort of samples, and it can have many applications, which goes through plant and animal trait selection, association with disease or resistant genes, sampling the population diversity and analysis of structure of those population and identification of adaptation patterns to understand how plants or crops are or can adapt over the climate changes that are occurring to our planet. And all of these applications need different level of resolution to cope with the goals. For example, we can run from just a phylogenetic analysis with a few hundreds of markers, while association studies might require hundreds of thousands, if not millions of markers. And so the flexibility of a genotyping platform is one key element to be able to provide a cost-effective solution to a quite wide range of scope of analysis.
There are basically two main platforms that have been seen running on the market in the last years, with the long run of array-based platform that have been proved to be a very robust system for acquiring genotyping data, and now in the recent years, sequence-based genotyping. And in this presentation, we are going to focus on a very specific solution from TICAN, which is the Allegro target genotyping. Despite the robustness of array-based platforms, we can make a comparison in favor of sequencing-based genotyping by several aspects. Array-based platforms are unmodifiable arrays, so once the array is designed and manufactured, it's very hard to implement with additional markers or scale up and down in the number of assayed polymorphism. It does require a specific instrumentation to be read out, and it also asks for a higher commitment in terms of sample number when the array is designed from scratch. And also the design per se requires a good SNP catalog to sample from to design the array. And that brings us to the concept of ascertainment bias, so the selective view of those arrays. So the array will be designed on a very specific set of polymorphisms, and only those polymorphisms are going to be genotyped through the experiments using that specific array. On the other side, sequence-based genotyping, especially the Allegro system, provides a modifiable system which can be changed over the time by means of the probes of the genes that are targeted. It leverages the ubiquity of NGS platform and their continuous cost decrease. It does not require a very high commitment in terms of sample number, and as well, the panel size can be really scalable from hundreds of loci up to hundreds of thousands of loci. And being a sequencing-based platform, it does remove the ascertainment bias problem. So every read will, along with the target polymorphism, whether those are selected, it will provide discovery and genotyping in the span of 100 or up to 300 base pair. Just to give you a picture on how the system works, so pretending that we have a known SNP that we want to target by our panel, probes that are basically primers, long primers, are designed upstream of that site, and they will provide priming to the extension event. That's why the technology is called SPET, single primer extension technology. And so those extensions will generate template molecules that are going to be then sequenced. And as you can see by the picture, yet you're going to extend over the target site, but you can also discover new sites that were not known before. That's all. It's all a measure on how long the reads are going to be. If they're going just to be 75 base pair, for example, you will probably just genotype mainly target SNPs. But if you extend your sequencing up to 250 base pair, you give a lot of room to de novo discovery and genotyping. The scheme of processing of Allegro genotyping is pretty simple. You work on plates, so on a single sample basis for the very first two steps of the procedure, which are fragmentation and ligation of adapters. Ligation of adapters carry the first molecular barcode that it's going to tag each individual sample within the plate. Then there is a pooling scheme on a two-tier approach, which leads you to having a single tube per plate. Then with that, on that single tube per plate, the enrichment, the single primer enrichment is performed. So you will enrich for the target loci,
And after that, a final PCR enrichment step is carried to provide the final libraries with a dual indexing scheme. So you basically introduce the second index, which will discriminate plates one another.
We have already published a couple of seminal papers on the technology. A first paper that came out on Annals of Botany and presented a comparative study of Allegro genotyping with Illumina SNP chip by using maize inbred lines and hybrids to compare the robustness of genotype calls. And another paper on Frontiers in Plant Science published by Barkey and co-worker, where we were involved in the genotyping of thousands of eggplant and tomato accessions. So I suggest you to give a look at those papers because they really prove the effectiveness of the technology in providing large genotyping datasets.
So we implemented this technology on a fully automated platform. Thanks to Tican, we have just recently installed the DreamPrep, which is this cool automatic liquid handler in our labs. It carries an on-deck thermocycler and a plate reader, so it's capable to carry all the NGS preparation without moving samples out of the deck. With the current scripting that we have in-house, we can process for the Allegro preparation 4 plates a day without moving anything out of the deck. So what we wanted to do was a stress test, and we prepare 564 samples divided in 6 plates of these maize magic lines. And the probe panel set that was used was a 50K design, actually selected from a previous pilot design. And those were designed on ZMI's assembly before. So what we did is for plate 1, everything was carried on the deck up to the pooling stage, and then we moved the rest of the processing outside of the deck, that is on the bench. For plate 2, plate 3, plate 4, and plate 6, we ran everything on the liquid handler or the DreamPrep. But for plate 3, where all the thermic cycling were carried on other thermocyclers. So that is the fragmentation, ligation, enrichment step, and final PCR were carried without using the on-deck thermocycler. And the plate 5 instead was completely processed on the benchtop. Here you can see the profile of the libraries. They are different in looking at the bioanalyzer, but finally when we align the data on the reference genome, you can see that all of them has a quite comparable fragment size. The only outlier here is plate 1, which, unlike the others, which had a median of about 300 base pair, plate 1 recorded a median fragment size of about 500 base pair. This is the plate that was processed half on the liquid handler and half on the benchtop. The first performance value that we wanted to assess was the level of enrichment across plates and preparation. As you can see here, regardless of whether plates were processed on deck, on the benchtop, or half away, the percentage of row reads that were finally informative in providing coverage over target loci was always in the range of 70 to 80%, which is quite of a very important result, because if you compare any other NGS technologies, I think you will find hard to see libraries with such a high amount of reads that are actually actionable reads for
the scope of the analysis. If we then look at the distributions of coverages across probes, we can see that for plates 1, 3, and 5, which are the plates that reach exactly what was intended to be the target coverage, we have a similar pattern of variation. We instead have a much higher median for plate 2, which was on purpose over-sequenced, while for plate 4 and 6, you can clearly see that they are lagging behind, and this is because we have not yet completed the sequencing for those. So please bear in mind that from now on, you should consider the entire dataset as not very complete, because we still need to get some additional coverage out of those 2 plates. Here in this big distribution, you can see a plotting of all the genotyping events accepted by a stratification of threshold from a minimum of 5x coverage up to a maximum of 30x coverage for each sample, given the amount of reads that have been generated for that sample. If we take as a first point of view a minimum coverage of 5x, which actually was enough for this experiment, because I recall that we are genotyping homozygous maize lines, so 5x is already a very robust call for an homozygous individual. Here is where you start to move from a poor genotyping cluster almost to a good level of saturation of the library is reached by having generated about 40 cluster, if you want 80 reads per probe per individual. While a much higher level of saturation, which can also be at that point considered valid for a minimum of coverage of 10x and 15x, is where 120 cluster per probe per sample are generated on the sequencer. And then you can consider here 200 cluster per probe as a complete saturation point, where regardless whether you are looking for genotyping at 5x or 30x, you are always on top of the recalling ratio. Of course, it's a trade-off of cost per data point there. We assume here to be already on a good spot when the number of cluster per sample per probe are within these 2 landmarks, ideally around 4 million reads. Sorry, 4 million fragments. Here it's a more detailed view if we split the data across plates. If we look only at data from plate 2, which is the over-sequenced plate, we can see that all the data points are completely saturated, and there is a very high coverage of genotyping, actually more than what is needed in terms of cost per data point. On the contrary, plate 4 and 6 are still in the slope of reaching a good saturation point, and that's why they need more sequencing data to have at least a median of 4 million fragments per individual. So what we have got so far is that out of this panel of 50,000 loci, in this magic population, we yielded more than 47,000 target sites to be polymorphic, and along with those, we also yielded more than 247,000 off-target sites being polymorphic. If we then put down some constraints and thresholds in the genotyping recall, by looking at those sites only genotyping a minimum of 384 samples, and yet you should consider that plate 4 and plate 6 are lagging behind in terms of coverages. With the minimum genotyping coverage of 3x, more than 43,000 target sites have been genotyped, And at the same coverage, more than 200,000 off-target sites, polymorphic, have been genotyped. Of course, if we increase the coverage to 10x, we see this number decreasing, not significantly, but we knew that for this type of population, being a nearly homozygous magic inbred lines, we don't need much than discovering the single haplotype being inherited in each line. So with this data, I leave the podium to Leonardo Caproni to continue on the analysis of these data points for GWAS and quantitative traits dissections. Thank you very much.
And thank you, Davide. I'd like to now welcome Leonardo for the second part of the webinar today. Welcome, Leonardo.
Thanks, Davide, and thanks all of you for attending this webinar. So I'm Leonardo Caproni. I work at Scuola Superiore Sant'Anna. And now, right before I tell you something about our case study, let me briefly introduce the group I work with at Scuola Superiore Sant'Anna. As you can see from the background of the picture, there's the Leaning Tower. Scuola Superiore Sant'Anna is located in Pisa. It is one of the 3 universities in the city, and it is an honors college. Here you can see the crop genetics group, and we like to work with cereals, and we explore genetic resources, and we like to work on quantitative genetics to develop tools for modern breedings. However, we also work a lot with landscape genomics, in the development of new breeding approaches for smallholder agriculture in developing nations, and we also work with multi-parental populations. So now we can start talking about multi-parental populations that are extraordinary materials to perform QTL mapping. However, before I start talking to you about QTL mapping, I thought it was a good idea to give you a brief recap on QTL mapping within plants, and I must mention mapping using biparental recombinant inbred lines populations. So here, as you can see in the picture, what's the principle behind QTL mapping in biparental populations is that you look at the diversity within a certain species. If you're looking for a specific trait, you should find 2 very divergent individuals for that trait. So what you do is you cross these 2 diverging parentals, P1 and P2 in the picture, then you obtain several 2-way hybrids, and then you start like selfing their progenies, and for 6 to 8 selfing generation, and what you do is you end up with a population of recombinant inbred lines that you can use to map your traits. However, this approach has several limiting factors. In fact, you may not be able to map other traits, as these populations are built upon a certain trait. So what you can do, you can also perhaps use association mapping, in which you take the whole or a very good representation of the diversity of the species you are working with, and you try to associate the phenotype you measure with this broad genetic diversity, but you totally ignore the parental information. You totally miss the pedigree. So to bridge the gap between mapping using biparental recombinant inbred population and association mapping, here we have multiparental population. The most popular and powerful designs are NUM and MAGIC. So for nested association mapping panels NUM, what you do is you basically produce a lot of biparental recombinant inbred line populations, but for each of these panels that you produce, you use a recurrent parent. Here is in this scheme is indicated in light blue. The other very popular um, multi-parental design is multi-parent advanced generation intercross, the so-called MAGIC populations. So what you do with MAGICs is basically you try to recombine the genomes of multiple parents. You can have 4, 8, 16 initial lines, and you end up basically with an inbred germplasm, which is actually the mosaic of the genomes of the initial parentals, and it constitutes an enduring resource to perform QTL mapping, as you can map many traits. So now we start talking about our magic mice. It is an 8-way magic that was first published in 2015. Here you see the multiple inbred founder lines that were crossed using a dialectic cross design, and after crossing the 2-way, 4-way hybrids, obtaining 8-way hybrids, and after several generations of selfing of single seed descent, we obtain a panel of reals that are mosaic of the founder genomes, and as you can see, we can find a lot of phenotypic diversity. Well, I told you it is an 8-way magic, but actually there is a 9th parent, which is called CML91, which was introduced upon the failure of the 2-way hybrid B96 by HP301, and was this hybrid was substituted with B96 by CML91 to compensate and to recover the failures of this cross. So why our magic population is such a powerful tool to perform QTL mapping? So here, from the work of 2015 of Matteo Dell'Acqua, here you can see we used this 529 magic lines, which were genotyped using a SNP BeadChip. We had circa 50,000 SNP markers, and we were able to map this QTL of Pollen Shed on chromosome 8. So nothing special. You see the peak. But however, if you combine these data with whole genome sequencing data and or transcriptomics data of the founder lines, you can go within your interval on your chromosome, and this information can help you in dissecting the genetic mechanism behind the trait, and you can start looking really at candidate genes. That's why these are great genetic resources to map QTL of any complex trait. Now we are using our magic mice within the framework of Capitalize. This is an Horizon 2020 funded project, and we're dealing with photosynthesis related traits. So we want to map photosynthesis related traits in 3 main crops, in maize, barley, and tomato. And to do so, we are using high throughput phenotyping facilities, as you can see in the picture. This is actually our magic mice population being phenotyped at IPK Gatos-Leben. So why Allegro genotyping for the magic mice? So within this framework of this project, we had the chance to produce new genotyping data on this population, and we choose Allegro technology because it's very important to target specific region, because you can capture the most rare and informative variants. Of course, if you have some preliminary information, I'm talking about whole genome sequencing of the founder lines of your population. Like this, you might be able to overcome the biases due to some array designs, as Davide was mentioning you before. So we decided to evaluate our new data sets by looking at the number of SNPs, passing a very strict QC, try to understand if we can overcome the bias due to the design, and we looked at SNP density and the distribution, and we finally were able to test the discovery potential of our new data set on a subset of samples of our population, around 300, through GWAS. Not QTL mapping, because still we need to produce more sequencing data, and after that we need to produce a genetic map, which still lacks actually. So we need to improve the coverage, and our aim today is to test the suitability of these data sets to move forward with QTL mapping. So if we compare Allegro genotyping with the BeadChip, yes, we were able to increase by threefold the number of SNPs, and we used for both dataset quite strict quality control parameters by sites and by individuals, as you can see here. And in order to compare the real number, what we did is we use a subset of samples which have both genotyping data and also a subset of reads that also have phenotypes. So next, we wanted to look at the ability of our new set of 160,000 SNP markers to avoid any bias due to marker selection based on a small set of elite genotypes. So within our founder lines, there are also exotic materials. As you may see from the two neighbor joining trees on the left, this one is represented using our new set of SNP markers, and this one on the right was produced using our old set of markers. Clearly, we can see that before, if you look at the right panel, we had quite limited ability to distinguish between some founder lines, and this made our life very hard in attempting to reconstruct the genomes of our recombinant inbred lines to perform QTL mapping. Most problem arose, you see here in red, between B96 and CML91, and between H99 and W153R. These biases are due on the fact that the BeadChip, we know, is mostly designed on other two parentals actually of this panel, which are B73 and NMO17, you see in dark red and light blue here. And this, of course, has like negative implications. In the left panel, however, we can observe a much more favorable situation, as we do not observe any bias. So our new marker set can definitely separate our founder lines, which makes us really happy for the downstream analysis in QTL mapping. SNP density matters, yes. Even if we got many more markers when we compare Allegro versus the BeadChip, we should also be careful and look at the distribution of our SNPs along each chromosome. Comparing the two datasets here you see in the picture, and looking at the average SNP density, here we are looking at a window of 200 kilobases, we can observe a much more favorable situation with the Allegro genotyping and a much higher density of SNPs, and this would help us a lot in the downstream analysis, especially in the attempt of reconstructing the genomes of our recombinant inbred lines to perform QTL mapping. Okay, so now we are ready to test finally whether our new genotyping dataset is good for the discovery of interesting genomic region linked to some of the phenotypes available. So as I mentioned before, we have a subset of circa 300 recombinant inbred lines that were genotyped with both methods. And these reels, of course, also underwent phenotypic characterization back in 2015. So what we did, we explored two GWAS using this method developed by Liu and colleagues that actually allows to prevent some model overfitting and controls false positives. So it's a quite refined regression for GWAS. And what we see here You see highlighted in red, we have the same old signal on chromosome 8, the one you saw on Pollen Shed. Here we are testing Pollen Shed. And but what we also see other 6 regions in chromosome 1, 2, 4, 5, and 6. And so it means that with these new data sets, we may unlock the discovery of new regions when we will perform QTL mapping. We did the same test using another very important phenotype, grain yield. And if we look at the data of 2015, here we see on chromosome number 6 here a very evident peak, and we see the same situation here with our new data sets. So these, of course, these are 2 different methods, but what we can see here, we got a significant signal highlighting the same chromosomic region on chromosome 6, but we see also a very interesting pattern in chromosome number 7. So even if we are using less samples, 300 here and more than 500 here, we are able to detect the same region. So we may have potential discovery of a new region in chromosome 7 here. So to conclude, I have to remark that for our side, using Allegro on DreamPrep gave us an increased statistical power. We have more SNPs, we have promising marker distribution, and this will help us a lot for our haplotype reconstruction, and we do not observe any bias. And we corroborate our previous QTL mapping results, and yes, we got evidence of potential discovery of new region even if we are using. So what's next? Within Capitalize, we still need to finalize our high throughput phenotyping experiments, and we need to develop a genetic map for our magic mice. Currently, we are refining our QTL mapping pipeline, and then we will be ready to perform our QTL analysis and map photosynthesis related traits. So, as final remarks on Allegro on DreamPrep, we must say that it removes pitfalls of array platforms. It has very competitive pricing per sample, as it leverages at most the continuous drop in costs of next generation sequencing. It is a flexible platform, and it also accepts continuous improvement. And very importantly, we must mention that it can become a community asset. And once you have a full panel, you just need a sequencing machine and you're ready to go. So Allegro technology and DreamPrep automation can bring reproducibility and speed to the next level. So before I finish, I want to acknowledge my group, especially Mario De Copè, Master dell'Acqua, and Maramikulan. I thank you all for your attention. Thanks.
And thank you so much, Davide and Leonardo, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. Gentlemen, it looks like we already have some questions coming in from our audience members. First question, why using targeted genotyping and not a WGS approach?
Okay, I think I can take this one. Targeted genotyping, I think it's the most valuable approach as long as the genome size is still big. And this is true for most of the agriculturally relevant crops. Whole genome sequencing can be a way to do genotyping, but it can beat on the cost side only if you're dealing with very small genomes like Arabidopsis or everything that is basically below 500 megabases. And yet you might see a convenience in going that way. But genotyping on a targeted way ensure you the ability to get very robust data point on very large cohorts, which is not that trivial also on a whole genome sequencing approach, because if you're going to do whole genome sequencing, I think you're going to opt for scheme sequencing, and there still you might get biases on the representation of the genome, and you increase the amount of missing data, and this can be highly detrimental in association studies. You can deal with missing data points, but you need to be able to impute those data points, and this is feasible only if you have a reference population to impute against. So to have clear data points, targeted genotyping is still strongly preferred, because whole genome sequencing will replace targeted genotyping only when the sequencing cost would be another order of magnitude less than the current cost.
Thank you very much. Now our next question is, which automation platform was used for the Allegro genotyping, and why was this approach chosen with TCAN?
As mentioned in the presentation, we use the DreamPrep, which is actually a specific setup of the Fluent. The Fluent is the latest automation from TCAN as liquid handler, and DreamPrep is the setup which I think is the middle-sized Fluent with incorporated thermocycler and the plate reader. We choose that one because, well, this is more of a company story. TCAN merged with Nugen Technologies, and Nugen Technologies formerly developed Allegro system, and now they are directly implementing this solution over their liquid handler. So we have quite of a straightforward support in implementing this system in our lab.
Thank you so much. And Leonardo, did you want to add to either one of those questions?
Yes, I would like to add something related to the first question that Davide answered. So why using target genotyping and not whole genome sequencing approaches? So looking at our population, I mean, like when you're using recombinant inbred lines, of course, it would be extremely good to perform whole genome sequencing on the whole set of recombinant inbred lines. But that would mean to invest a lot of money, because actually the genome of mice is extremely big. So I'm talking about our situation. It's around 2.4 gigabases. So the idea and the advantages of this population is that you can perform targeted genotyping on the recombinant inbred lines, and perhaps you may produce whole genome sequencing on the founder lines, and once you are able and you to reconstruct the mosaics within each of the chromosomes related to the genomes of the founder, these mosaics can then be inferred from SNPs. And then you can infer with whole genome sequencing actually the real condition within those chromosomic regions. I don't know, perhaps I have been a little bit confusing, but the idea is that you can impute the real state within a certain chromosomic region using the whole genome sequencing data of your founder lines. So there is no need to use whole genome sequencing on recombinant inbred lines. this is the point I wanted to tell.
Thank you so much for adding that piece. And gentlemen, how many samples can be prepared for sequencing in a working day?
Well, depends on if you implement automation in your lab or not. In our lab, basically, with automation, we have been able to process up to 8 plates per day. It's a matter of working on the deck or not. I mean, you can parallelize more if you have a battery of PCR machines to do the PCR steps or the incubation step. Well, actually, the incubation step is not limited by any platform at all. But that's the point. So far in our lab, given our needs, we have been able to run 8 plates per day, which is already quite a sizable amount of samples. But I know from Tecan that they have also developed combined solution involving multiple liquid handler doing separate steps of the process, and they can scale up to multiples of the 8 plates we're doing here with a single robot.
Wonderful, thank you very much. And I want to thank our audience for these great questions coming in. We have time for a few more. What is the advantages of using recombinant inbred lines for identifying regions for the genome linked to traits?
Okay, perhaps I made like comment on this question. So actually, the advantage is that like using the pedigree information of each of your recombinant inbred line, you can actually map really precisely in a certain chromosomic region your trait of interest. And what you can do with these populations, once you identify a certain chromosomic region, you can go further by looking at and performing association mapping with the whole genome sequencing data of your founder lines within that specific region. And what you can also do with recombinant inbred lines population, with populations such as our magic mice population, you can also use RNA sequencing data and look at the pattern of expression of genes within that region. So what you can basically do, like more than association mapping, is that you can really look into a certain defined chromosomic region, and you can really dissect the mechanism behind the trait you are studying. This is the true advantage. And of course, using this population, you can use and reuse all the genotyping and phenotyping data in the years to come. I mean, it's an indeterminate resource, and of course, this has several advantages in terms of costs and in terms of adding quality to the research you carried out in the past. You may add further information in the future. Okay, thanks.
Thank you so much, gentlemen. We have time for 2 more questions. Our next question, how does Allegro compare to genotyping by sequencing?
I will take this one. This is actually a very important question for those that are not familiar with targeted genotyping. Standard genotyping by sequencing is based on restriction enzymes. And there you randomly sample the genome without any specific target to be selected. It was a very good first step in using the genotyping, using sequencing as a genotyping platform. However, it has several limitations. One of those is that if you have a big genome and you want to find association for phenotypic traits, usually those mutations reside within, they reside within or close by genes. And so you want to yield genotyping data points that are in genes or at least nearby genes. And if you randomly sample, let's say 30, 50,000 loci using standard GWAS technologies, you don't get the statistical power because you are too far from the causative mutation, and the linkage decay will cause you the loss of complete loss of the statistical power. This is one aspect. The second aspect is that GWAS is hardly reproducible, because the selection of loci of the genome that you are going to take in the sequencer strongly depends on, of course, the restriction enzyme that you can reproduce as long as you know the protocol, but also the size selection is a very pivotal point in the procedure, and whether you select for small fragment or large fragment, you change the population of loci. So it's very hard to reproduce across batches if you don't have a very robust protocol, and very, very hard to reproduce across laboratories. While instead, like Allegro genotyping, once the probe set has been designed and shared, it will work the same way across several labs and several operators the same way, and you keep having recall across the same loci, yet on top of interesting gene. Targeted genotyping can also be used to focus your genotyping not across the entire genome, but maybe to do fine mapping on a specific chromosome arm. So you have complete flexibility on choose where you want to get your genotyping data points. And not talking how useful can be for extremely huge genomes such as wheat or gymnosperms.
Thank you so much. We have time for one more question, gentlemen. What are the benefits for this genotyping approach to be standardized, and how can this be done?
The benefits, of course, is to try to develop, to democratize the genotyping in that sense, like Leonardo was mentioning before. Now the sequencing capabilities are going to be a much more distributed asset across labs, academies, research institutes, and so on. So the proximity to a sequencer is going to be much more democratic rather than a genotyping facility. And Allegro genotyping is quite of an example where as the probe set, the design of a specific panel is shared and independently acquired by many labs, those many labs have the capability to reproduce exactly the same experiment because, well, the protocol is fixed, and you can get an extra layer to ease the process by having the automation. But even if you do it by hand on the benchtop, it's still a reproducible protocol, and then the loci that are going to be genotyped are going to be exactly the same of those done in another lab. And this can be a facilitator on building on communities of on genetic resources in general.
Gentlemen, thank you so much for your presentation today and your time. Would you like to provide any closing remarks to our audience before we go?
I think that one effort one should do when considering developing arrays is the number of samples that are going to be genotyped and expected coverage. Like I was mentioning before,
because this is a pivotal factor that we are on a daily basis consulting with our clients to decide the amount of coverage and to make the experiment the more cost-effective as possible. Also, I want to mention that targeted genotyping is worth doing when you have to do at least, let's say, a couple of plates of individuals. Otherwise, the design process it's more costly than the saving %.
Gentlemen, I want to thank you again for your time today and for your important research. And I also want to thank LabRoots and our sponsor TCAN for underwriting today's educational webcast. And before we go, of course, I want to thank our audience members for joining us today and for their interesting questions and questions we did not have time for today, and those submitted during on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you again. Take care. Bye-bye.